Heredity and evolution. Introduction. Heredity is the transmission of characteristics, physical or mental, from parents to offspring, that is, from one generation to the successive generation. It is of common knowledge that like produces like. Human beings give birth to babies, a cat gives birth to kittens, and a hen produces chicks. Although offsprings resemble their parents, they are not identical to them. They exhibit a departure in some of the qualities from the previous generations. These differences shown by the individuals of a species are termed as variations. Thus, variation means the differences that exist in the individuals of a species. It represents the differences that permits distinction between two individuals of the same race or between the offsprings of the same parents. The scientific study of the transmission of hereditary character from one generation to the other is known as genetics. If we look around us, we see different kinds of organisms. The organisms are of different species. Even among the species, there are differences. For example, one cat is different from the other. One lion is different from the other. And also, human beings are different from each other. Except for identical twins, even offspring born to same parents are different. All these differences are classified as variations. Both heredity and variation play important roles in evolution of organisms. Accumulation of variation during reproduction. Inheritance from the previous generation provides both a common basic body design and subtle changes in it for the next generation. Now think about what would happen when this new generation in its turn reproduces. The second generation will have differences that they inherit from the first generation, as well as newly created differences. If one bacterium divides and then the resultant two bacteria divide again, the four individual bacteria generated would be very similar. There would be only very minor differences between them, generated due to small inaccuracies in DNA copying. However, if sexual reproduction is involved, even greater diversity will be generated, as we will see when we discuss the rules of inheritance. Do all these variations in a species have equal chances of surviving in the environment in which they find themselves? Obviously not. Depending on the nature of variations, different individuals would have different kinds of advantages. Bacteria that can withstand heat will survive better in a heat wave, as we have discussed earlier. Selection of variants by environmental factors forms the basis for evolutionary processes. Heredity. The most obvious outcome of the reproductive process still remains the generation of individuals of similar design. The rules of heredity determine the process by which traits and characteristics are reliably inherited. Let us take a closer look at these rules. Inherited traits. What exactly do we mean by similarities and differences? We know that a child bears all the basic features of a human being. However, it does not look exactly like its parents, and human populations show a great deal of variation. Rules for the inheritance of traits. Mendel's contributions. The rules for inheritance of such traits in human beings are related to the fact that both the father and the mother contribute practically equal amounts of genetic material to the child. This means that each trait can be influenced by both paternal and maternal DNA. Thus, for each trait, there will be two versions in each child. What will then the trait seen in the child be? Mendel worked out the main rules of such inheritance, and it is interesting to look at some of his experiments from more than a century ago. Mendel used a number of contrasting visible characters of garden peas, round, wrinkled seeds, tall, short plants, white, violet flowers, and so on. He took pea plants with different characteristics. A tall plant and a short plant, produced progeny from them, and calculated the percentages of tall or short progeny. In the first place, there were no halfway characteristics in the first generation, or F1 progeny, no medium height plants. All plants were tall. This meant that only one of the parental traits was seen, not some mixture of the two. So, the next question was, were the tall plants in the F1 generation exactly the same as the tall plants of the parent generation? Mendelian experiments test this by getting both the parental plants and these F1 tall plants to reproduce by self-pollination. The progeny of the parental plants are, of course, all tall. However, the second generation or F2 progeny of the F1 tall plants are not all tall. Instead, 1/4 of them are short. This indicates that both the tallness and shortness traits were inherited in the F1 plants, but only the tallness trait was expressed. Thus, two copies of the trait are inherited in each sexually reproducing organism. These two may be identical or may be different depending on the parentage. A pattern of inheritance can be worked out with this assumption as shown in the figure. In this explanation, both capital T, T and capital T and small t are tall plants, while only small t is a short plant. In other words, a single copy of the capital T is enough to make the plant tall, while both copies have to be the small t for the plant to be short. Traits like the capital T are called dominant traits, while those that behave like the small t are called recessive traits. Work out which trait would be considered dominant and which one recessive in the figure. What happens when pea plants showing two different characteristics rather than just one are bred with each other? What do the progeny of a tall plant with round seeds and a short plant with wrinkled seeds look like? They are all tall and have round seeds. Tallness and round seeds are thus dominant traits. But what happens when these F1 progeny are used to generate F2 progeny by self-pollination? A Mendelian experiment will find that some F2 progeny are tall plants with round seeds and some were short plants with wrinkled seeds. However, there would also be some F2 progeny that showed new mixtures. Some of them would be tall but have wrinkled seeds, while others would be short but have rounded seeds. Thus, the tall-short trait and the round seed, wrinkled seed trait are independently inherited. Another example is shown in the figure. Sex determination. We have discussed the idea that the two sexes participating in sexual reproduction must be somewhat different from each other for a number of reasons. How is the sex of a newborn individual determined? Different species use very different strategies for this. Sex determination. We have discussed the idea that the two sexes participating in sexual reproduction must be somewhat different from each other for a number of reasons. How is the sex of a newborn individual determined? Different species use very different strategies for this. Some rely entirely on environmental cues. Thus, in some animals, the temperature at which fertilized eggs are kept determines whether the animals developing in the eggs will be male or female. In other animals, such as snails, individuals can change sex, indicating that sex is not genetically determined. However, in human beings, the sex of the individual is largely genetically determined. In other words, the genes inherited from our parents decide whether we will be boys or girls. But so far, we have assumed that similar gene sets are inherited from both parents. The explanation lies in the fact that all human chromosomes are not paired. Most human chromosomes have a maternal and a paternal copy, and we have 22 such pairs. But one pair called the sex chromosomes is odd in not always being a perfect pair. Women have a perfect pair of sex chromosomes, both called X. But men have a mismatched pair in which one is a normal-sized X while the other is a short one called Y. So women are XX while men are XY. All children will inherit an X chromosome from their mother, regardless of whether they are boys or girls. Thus, the sex of the children will be determined by what they inherit from their father. A child who inherits an X chromosome from her father will be a girl, and one who inherits a Y chromosome from him will be a boy. Evolution. We have noted that there is an inbuilt tendency to variation during reproduction, both because of errors in DNA copying and as a result of sexual reproduction. Let us now look at some consequences of this tendency. An illustration. Consider a group of 12 red beetles. They live, let us assume, in some bushes with green leaves. Their population will grow by sexual reproduction and therefore can generate variations. Let us imagine also that crows eat these beetles. The more beetles the crows eat, the fewer beetles are available to reproduce. Now let us think about some different situations that can develop in this beetle population. In the first situation, a color variation arises during reproduction so that there is one beetle that is green in color instead of red. This beetle, moreover, can pass the color on to its progeny so that all its progeny beetles are green. Crows cannot see green-colored beetles on the green leaves of the bushes and therefore cannot eat them. In a second situation again, a color variation arises during reproduction, but now it results in a beetle that is blue in color instead of red. The beetle can also pass the color on to its progeny so that all its progeny beetles are blue. Crows can see blue-colored beetles in the green leaves of the bushes as well as they can see red ones and therefore can eat them. What happens initially? In the population as it expands, there are a few blue beetles, but most are red. But at this point, an elephant comes by and stamps on the bushes where the beetles live. This kills most of the beetles. By chance, the few beetles that have survived are mostly blue. The beetle population slowly expands again, but now the beetles in the population are mostly blue. It is obvious that in both situations, what started out as a rare variation came to be a common characteristic in the population. In other words, the frequency of an inherited trait changed over generations. Since genes control traits, we can say that the frequency of certain genes in a population changed over generations. This is the essence of the idea of evolution. Speciation. What we have seen so far is microevolution. That means that the changes are small even though they are significant. Also, they simply change the common characteristics of a particular species, but this does not properly explain how new species come into existence. That can be said to have happened only if this group of beetles we are thinking about splits into two populations that cannot reproduce with each other. When this happens, they can be called two independent species. So, can we extend the reasoning we have used above to explain such speciation? Consider what would happen if the bushes the beetles feed on are spread widely over a mountain range. The beetle population becomes very large as a result, but individual beetles feed mostly on a few nearby bushes throughout their lifetime. They do not travel far. So, in this huge population of beetles, there will be subpopulations in neighborhoods. Since male and female beetles have to meet for reproduction to happen, most reproduction will be within these subpopulations. Of course, an occasional adventurous beetle might go from one side to another. Or a beetle is picked up by a crow from one side and dropped in the other side without being eaten. In either case, the migrant beetle will reproduce with the local population. This will result in the genes of the migrant beetle entering a new population. This kind of gene flow is bound to happen between populations that are partly but not completely separated. If, however, between two such subpopulations, a large river comes into existence, the two populations will be further isolated. The levels of gene flow between them will decrease even further. Over generations, genetic drift will accumulate different changes in each subpopulation. Also, natural selection may also operate differently in these different geographic locations. Thus, for example, in the territory of one subpopulation, crows are eliminated by eagles. But this does not happen for the other subpopulation, where crow numbers are very high. As a result, the green variation will not be selected at the first site, while it will be strongly selected at the second. Together, the process of genetic drift and natural selection will result in these two isolated subpopulations of beetles becoming more and more different from each other. Eventually, members of these two groups will be incapable of reproducing with each other even if they happen to meet. There can be a number of ways by which this can happen. If the DNA changes are severe enough, such as a change in the number of chromosomes, eventually the germ cells of the two groups cannot fuse with each other. Or a new variation emerges in which green females will not mate with red males, but only with green males. This allows very strong natural selection for greenness. Now, if such a green female beetle meets a red male from the other group, her behavior will ensure that there is no reproduction between them. Effectively, new species of beetles are being generated. Evolution and classification. Based on these principles, we can work out the evolutionary relationships of the species we see around us. It is a sort of going backwards in time. We can do this by identifying hierarchies of characteristics between species. In order to understand this process, let us think back to our discussion on the classification of organisms in Class 9. Similarities among organisms will allow us to group them and then study the groups. For this, which characteristics decide more fundamental differences among organisms and which one decides less basic differences? What is meant by characteristics anyway? Characteristics are details of appearance or behavior, in other words, a particular form or a particular function. That we have four limbs is thus a characteristic. That plants can do photosynthesis is also a characteristic. Some basic characteristics will be shared by most organisms. The cell is the basic unit of life in all organisms. The characteristics in the next level of classification would be shared by most but not all organisms. A basic characteristic of cell design that differs among different organisms is whether the cell has a nucleus. Bacterial cells do not, while the cells of most other organisms do. Among organisms with nucleated cells, which ones are unicellular and which ones multicellular? That property marks a very basic difference in body design because of specialization of cell types and tissues. Among multicellular organisms, whether they can undertake photosynthesis or not will provide the next level of classification. Among the multicellular organisms that cannot do photosynthesis, whether the skeleton is inside the body or around the body will mark another fundamental design difference. We can see that even in these few questions that we have asked, a hierarchy is developing that allows us to make classification groups. The more characteristics two species will have in common, the more closely they are related. And the more closely they are related, the more recently they will have had a common ancestor. An example will help. A brother and a sister are closely related. They have common ancestors in the first generation before them, namely their parents. A girl and her first cousin are also related, but less than the girl and her brother. A girl and her first cousin are also related, but less than the girl and her brother. This is because cousins have common ancestors, their grandparents, in the second generation before them, not in the first one. We can now appreciate that classification of species is in fact a reflection of their evolutionary relationship. We can thus build up small groups of species with recent common ancestors than supergroups of these groups with more distant common ancestors and so on. In theory, we can keep going backwards like this until we come to the notion of a single species at the very beginning of evolutionary time. If that is the case, then at some point in the history of the earth, non-living material must have given rise to life. There are many theories about how this might have happened. It would be interesting to come up with theories of our own. Tracing evolutionary relationships. When we try to follow evolutionary relationships, how do we identify characteristics as common? These characteristics in different organisms would be similar because they are inherited from a common ancestor. As an example, consider the fact that mammals have four limbs, as do birds, reptiles, and amphibians. The basic structure of the limbs is similar, though it has been modified to perform different functions in various vertebrates. Such a homologous characteristic helps to identify an evolutionary relationship between apparently different species. However, all similarities simply in organ shape are not necessarily because of common ancestry. What would we think about the wings of birds and bats, for example? Birds and bats have wings, but squirrels and lizards do not. So are birds and bats more closely related to each other than to squirrels or lizards? Before we jump to this conclusion, let us look at the wings of birds and bats more closely. When we do that, we find that the wings of bats are skin folds stretched mainly between elongated fingers. But the wings of birds are a feathery covering all along the arm. The designs of the two wings, their structure and components are thus very different. They look similar because they have a common use for flying, but their origins are not common. This makes them analogous characteristics rather than homologous characteristics. It would now be interesting to think about whether bird arms and bat arms should be considered homologous or analogous. Fossils. Such studies of organ structure can be done not only on current species but also on species that are no longer alive. How do we know that these extinct species ever existed? We know this from finding fossils. What are fossils? Usually, when organisms die, their bodies will decompose and be lost. But every once in a while, the body or at least some parts may be in an environment that does not let it decompose completely. If a dead insect gets caught in a hot mud, for example, it will not decompose quickly and the mud will eventually harden and retain the impression of the body parts of the insect. All such preserved traces of living organisms are called fossils. How do we know how old the fossils are? There are two components to this estimation. One is relative. If we dig into the earth and start finding fossils, it is reasonable to suppose that the fossils we find closer to the surface are more recent than the fossils we find in deeper layers. The second way of dating fossils is by detecting the ratios of different isotopes of the same element in the fossil material. It would be interesting to find out exactly how this method works. Evolution by stages. A question that arises here is if complicated organs such as the eye are selected for the advantage they provide, how can they be generated by a single DNA change? Surely such complex organs will be created bit by bit over generations. But how can each intermediate change be selected for? There are a number of possible explanations. Even an intermediate stage such as a rudimentary eye can be useful to some extent. This might be enough to give a fitness advantage. In fact, the eye, like the wing, seems to be a very popular adaptation. Insects have them, so does an octopus, and so do vertebrates. And the structure of the eye in each of these organisms is different enough for them to have separate evolutionary origins. Also, a change that is useful for one property to start with can become useful later for quite a different function. Feathers, for example, can start out as providing insulation in cold weather, but later they might become useful for flight. In fact, some dinosaurs had feathers, although they could not fly using the feathers. Birds seem to have later adapted the feathers to flight. This of course means that birds are very closely related to reptiles since dinosaurs were reptiles. It is all very well to say that very dissimilar looking structures evolved from a common ancestral design. It is true that analysis of the organ structure in fossils allows us to make estimates of how far back evolutionary relationships go. But those are guesses about what happened in history. Are there any current examples of such a process? The wild cabbage plant is a good example. Humans have over more than 2000 years cultivated wild cabbage as a food plant and generated different vegetables from it by selection. This is of course artificial selection rather than natural selection. So some farmers have wanted to select for very short distances between leaves and have bred the cabbage we eat. Some have wanted to select for arrested flower development and have bred broccoli, or for sterile flowers and have made the cauliflower. Some have selected for swollen parts and come up with kohlrabi. Some have simply looked for slightly larger leaves and come up with a leafy vegetable called kale. Would we have thought that all these structures are descended from the same ancestor if we had not done it ourselves? Another way of tracing evolutionary relationships depends on the original idea that we started with. That idea was that changes in DNA during reproduction are the basic events in evolution. If that is the case, then comparing the DNA of different species should give us a direct estimate of how much the DNA has changed during the formation of these species. This method is now extensively used to define evolutionary relationships. Evolution should not be equated with progress. In an exercise of tracing the family tree of species, we need to remember certain things. Firstly, there are multiple branches possible at each and every stage of this process. So, it is not as if one species is eliminated to give rise to a new one. A new species has emerged. But that does not necessarily mean, like the beetle example we have been thinking about, that the old species will disappear. It will all depend on the environment. Also, it is not as if the newly generated species are in any way better than the older one. It is just that natural selection and genetic drift have together led to the formation of a population that cannot reproduce with the original one. So, for example, it is not true that human beings have evolved from chimpanzees. Rather, both human beings and chimpanzees have a common ancestor a long time ago. That common ancestor is likely to have been neither human nor a chimpanzee. Also, the first step of separation from that ancestor is unlikely to have resulted in modern chimpanzees and human beings. Instead, the two resultant species have probably evolved in their own separate ways to give rise to the current forms. In fact, there is no real progress in the idea of evolution. Evolution is simply the generation of diversity and the shaping of the diversity by environmental selection. The only progressive trend in evolution seems to be that more and more complex body designs have emerged over time. However, again, it is not as if the older designs are inefficient. So many of the older and simpler designs still survive. In fact, one of the simplest life forms, bacteria, inhabit the most inhospitable habitats like hot springs, deep-sea thermal vents, and the ice in Antarctica. In other words, human beings are not the pinnacle of evolution, but simply yet another species in the teeming spectrum of evolving life. Human evolution. The same tools for tracing evolutionary relationships, excavating, time dating, and studying fossils as well as determining DNA sequences have been used for studying human evolution. There is a great diversity of human forms and features across the planet, so much so that for a long time people used to talk about human races. Skin color used to be the commonest way of identifying these so-called races. Some were called yellow, some black, white or brown. A major question debated for a long time was, have these apparent groups evolved differently? Over recent years, the evidence has become very clear. The answer is that there is no biological basis to the notion of human races. All humans are a single species. Not only that, regardless of where we have lived for the past few thousand years, we all come from Africa. The earliest members of the human species, Homo sapiens, can be traced there. Our genetic footprints can be traced back to our African roots. A couple of 100,000 years ago, some of our ancestors left Africa while others stayed on. While the residents spread across Africa, the migrants slowly spread across the planet from Africa to West Asia, then to Central Asia, Eurasia, South Asia, and East Asia. They traveled down the islands of Indonesia and the Philippines to Australia, and they crossed the Bering Land Bridge to the Americas. They did not go in a single line, so they were not traveling for the sake of traveling, obviously. They went forwards and backwards, with groups sometimes separating from each other, sometimes coming back to mix with each other, even moving in and out of Africa. Like all other species on the planet, they had come into being as an accident of evolution and were trying to live their lives the best they could.
Yes, I've been living inside of you. But I guess this time I set me free, and there's nothing that you can do. I hope you can see.
I can't seem to keep people around me, cause I'm scared that they might know the truth all about me. Then they might doubt me, there's no other route. I wanted to shout, but it's too late now, cause I'm drowning. Got two-faced like I'm a clown, man. Life is too great, no, it's astounding. I'm in good shape, ma, but you know that I'm drowning like a cup in a cage. I can't cope with this hopelessness, though I try to be open and go with it. But I'm soaking in the pain like the wind never stop flowing in. As I'm floating in a glowing river through a golden gate, trying to motivate. Yeah, I do my fucking best, but there's two voices in my head, I just can't. I just fucking can't.
Cause I've been living inside of you, but I guess this time I set me free, and there's nothing that you can do. I hope you can see me, cause I've been living inside of you, but I guess this time I set me free, there's nothing that you can do. I hope you can see.
Babe, what you acting like this? You be fine, trust me, just in time we get along. No, leave me alone. Why yes, can you see that you are alone, big? We're now sitting in our own throne, big. We need nobody, can you see it through? But it's not you who I should be listening to. Guess what, big? Nobody fucking needed you. No, and nobody fucking needed me. Yeah, they never needed us. Like when they nailed Jesus up, there's no fucking heaven that they don't believe in us. But they my friend, I'm the only one that you can trust. Yeah, god damn it, you an idiot. Go online, be a slave for the media. And you forgot what your father said. Are you serious? Look at yourself, big. You fucking idiots.
Cause I've been living inside of you, but I guess this time I set me free.
Oh, I love you.
And there's nothing that you can do.
But the guy is always, I see.
I know you can see.
Can you see it, baby? There's no one else.
Cause I've been living inside of you.
You have to accept it.
But I guess this time I set me free. And there's nothing that you can do.